To start with me a bit, I am Victoria Nugnes. My training is on biotechnology. I'm a PhD in science and technology. I'm Argentinian. I study at the University of Quilmes in Argentina, but now I have my position at the University of Padova, as Maria Cristina was saying, at the Biocompetitive Lab. I have a postdoctoral researcher, and I'm acting as the lead curator of this database. I'm also part of the International Society of Biocuration as an executive member. It's an international society that promotes biocuration and standardized information. I am working for Italy, but I'm based in Argentina. So for those of you that are also based in Argentina and are worried about the time difference, don't be. You can reach out to me in Europe time or Argentinian time. It's perfectly fine. So this is a bit of a layout of what the workshop is going to be about. Firstly, we're going to be doing a bit of an overview of what DISPROT and IDPs are. We're going to explore DISPROT, go through DISPROT annotations and ontologies, an overview of the curation process, the annotation of structural states, transitions, and functions of intrinsically disordered regions. What is MIADE? We're going to be seeing a bit about the region process and the crediting through API-Curon, which is an external platform, how we do curation in DISPROT, and how we do our trainings, a curation example, and a mock curation case, if the time allows us. We're going to see how we go on time. If we have the time, I would love to do a mock curation case, but that's sometimes not the case. If we don't get to do it today, I'm going to be sharing a video with you about how we curate an actual publication. So you have all the materials and all the knowledge necessary for you to become a DISPROT curator, okay? Once again, and before starting, if you don't understand, I talk too fast. I have a really strong Spanish accent. So if you have any issues with understanding what I'm saying or what I'm trying to say, please raise your hand, intervene. You are not bothering us. You are just helping for the training to go way better. A couple of days ago, I heard that when one person is asking, there's like 10 more people that are actually wondering about that. So be the brave one, bring your questions, and let's make this course really fun, okay? Or light, at least. So let's go to the overview of DISPROT and IDPs. DISPROT is the major repository of manually created data about intrinsically disordered proteins and intrinsically disordered regions, IDPs or IDRs. It covers or relies on experimentally validated evidences about the features and characteristics of these proteins. So we rely on evidence that are as a result of experiments done in wet labs, okay? Here you have a screenshot of, I don't know if you can see my mouse, but here we have some screenshots of our latest publication. You are more than welcome to check them, to know more about how we do things, how DISPROT is evolving, and the best practices for the manual creation of intrinsically disordered proteins in DISPROT. I think we also add them in the Google Drive that you should all have access, but in any case, if you don't find it or whatever, they are also linked here. So this is how DISPROT homepage looks like. Let's just do the laser. But what are IDPs? I hope all of you could go through the introductory material if you're not already familiar with what intrinsic disorder is. We are not going to cover that too much in this stage, but I'm going to introduce what they are very briefly. So we all know globular proteins. This is an example on the left. It has secondary structure features and even tertiary structure features. On the right side, we have examples of what intrinsic disorder is. Intrinsic disorder could present as local disorder or even global disorder. So this means that It could be just a portion of the protein, or it could be the whole protein. And the thing is that the IDPs, or intrinsically disordered regions, are those that lack this fixed 3D structure, okay? They might lack secondary and or tertiary structures. So they're very flexible and they move around a lot. And this is why they're represented here as an ensemble of different conformations. Here you have listed some interesting publications to know more about intrinsic disorder. So if you want to check them, you're more than welcome. So basically, if we were to define intrinsically disordered proteins or regions based on their nature, we will say that they do not form 3D structures that are stable, okay? Based on their composition, we can say that they are enriched in charge and hydrophilic residues. This is what prevents them to form like a core hydrophobic core. And this is what makes them very flexible and very elongated as well. Regarding the functional aspects of IDPs, contrary to the past views when the structure was related to the activity and was thought that if the proteins didn't have a structure, didn't have an activity, we now know that the proteins that are intrinsically disordered are very active and are involved in many cellular functions. Because they are so flexible, they are very promiscuous in their interactions, so they can facilitate the interactions through a variety of different partners. If we look into the roles, they are very well represented in signaling processes, in regulation, and in a variety of other cellular processes as well. Also, because they are so promiscuous, they are oftenly the cells oftenly try to regulate their activity by modifying them through post-translation and modification. So it's very common to find them as subjects of PTMs. Is there any question so far about what IDPs are or the features that they have? Okay, awesome. Also, I wanted to ask you, I forgot to mention this before, but if you are available, if you can, because I know it's not possible, but if you can turn on your camera, it would be really good for me because I can see your non-verbal cues, like nodding or your confusion. If it's not possible because you are sharing the space with someone else, it's completely fine and understandable, but if you can, it would be great. Thank you. So let's move ahead then. Very flexible. Let's summarize the key physicochemical features of the intrinsic disorder. They are highly mobile, they're highly flexible, they have like an elongated shape, they have a particular amino acid composition that is enriched in charged hydrophilic residues. So all of these features are able to be assessed by a different and variety of experimental approaches. And these are the approaches that we in this project focus on when we are trying to add information about these proteins, because these are the different kinds of information that we can retrieve from these proteins. These include, but not just rely on NMR experiments, nuclear magnetic resonances, mass experiment, SACS, circular dichroism, and many, many others. There are other experiments that don't necessarily point directly or evidence disorder itself, but could point towards disorder adjacent evidence, like X-ray crystallography and also cryo-electronic microscopy as well. These are experiments that we rely a lot on as well. If you want to know more about different experimental approaches, you have them on their Google Drive folder, some publications, and also you have here a list of publications that could be very useful. So let's go ahead and start exploring this spread, okay? As I told you before, this is what you will see if you go into disprot.org. This is our homepage. And you can find at the top navigation tabs that could lead you to the browse, ontology, variety of different stages of the page. You have the search box where you can type the protein names or whatever you feel like searching. examples of well-annotated entries or very oftenly consulted entries, list of organisms, and list of thematic datasets. In particular, when we look at the portion of the organisms, there are the collection of the different entries or proteins that we have that relate to the model organisms that you see in the screen. This doesn't mean that in this plot we don't have other organisms, but since these are the model organisms, they are the most consulted ones, so that's why they're highlighted in the web page. The number here represents the number of proteins that we have on this plot for the disorder proteins that belong to those organisms. And then we have the data sets. These are collection of proteins that are related to a particular biological area of relevance. They could be related to diseases like cancer-related proteins, or features like RNA binding proteins, or things like bacterial virulence-related proteins. You can see we have extracellular matrix protein and a variety of thematic data sets. We try to focus on different thematic data sets. Every release that we do releases every 6 months. And when we do this, we also collect or create a dedicated blog post that you can find also linked in the homepage. You can find information about the data sets that we were working on, but also find information about courses or training materials that are new or are newly available at least. So how can I find if a protein or a publication is annotated or is present in this project? One of the things that you could do is use the search box, as I was telling you before, on the homepage, just typing in here the protein. But another option is go to the Browse tab and do a more granular search using the different fields that we have available. You can select protein name, UniProt accession number, dysprot ID, organism, whatever. If you were to select, for example, protein name, you can type the protein name, let's say nucleoprotein. And dysprot is going to retrieve all the entries or proteins that has nucleoprotein as part of the protein name. It's also available to retrieve the information by the download. You will have a button here, and you can download the information in a variety of different formats: JSON, CSV, FASTA, and GAF formats. And you can also perform a more specific search. You can add other fields, and let's say you now want to filter by organism as well, and you can select organism. You can just type severe, and you will be retrieving the protein from the nucleoprotein from SARS-CoV-2. Okay, so I'm using entry and protein as synonyms, and that's because that's what it is. It's a synonym. So let's see what happens if we go into an entry or a protein in this plot. This is what we will see. This is not nucleoprotein, this is a different one, but it's the same for each and every one of the proteins that we have. As you can see, we have different paths of what the entry has. At the top, we will see the protein details. We'll see the dysprot identifier. We get one identifier for each and every one of the different proteins that we have there. So we have one single identifier for the information that we have. We have the protein details, as I was saying, regarding the organism, the gene symbol, the sequence length, the disorder content, that this is related to the annotations about disorder that we have, the information about disorder that we have for that protein itself. Other cross-data databases references, and whether this protein belongs to a particular thematic data set. Then we'll see, this is called feature viewer, and it's like a schematic representation of the protein. What you see here at the bottom of the feature viewer is the representation of the whole length of the protein. So if we look into it more closely, we're going to find that it's also divided in two. We see that the top portion of it is related to the annotations or the information that dysprot has on this protein. And then we have some external resources track that relates to like interpro or AlphaFold to retrieve information that is like the domains of the protein for the case of interpro, or even for AlphaFold, the prediction of disorder regions based on the PLDDT score. So when you are seeing the reddish and even the yellowish portions of the protein are trying to say that the AlphaFold could not predict a structure there. So it's likely, or we could be tempted to think it's disorder, okay? But again, by a predictor. All the things that are annotated and the things that we have evidences, strong evidences for, are at the top of the feature viewer and are related, again, to our annotations. As you can see, there are different colors here because we have different types of annotations. We add information about the structural state, what state regarding the structure, aka is it disorder or is it not disorder, is the protein region. We also annotate transitions. You will see them as purplish in the feature viewer, and functions that are performed or involve these regions in reddish. Okay? So after the feature viewer, we're going to see this is a cropped image, but each and every one of these annotations, of pieces of information, are related to annotations that you are going to find below the feature viewer. This is how an annotation looks like. Annotation is a term that we use to say a piece of evidence, a piece of information that relates to this protein. So let's explore what a piece of evidence or an annotation looks like. An annotation, again, is a curated information about the disorder protein region. Something that I didn't say, but I'm going to try to highlight and stress as much as possible. DISPROT is a database about disorder proteins. We care about the disorder regions that are within the protein, whether they are fully disordered or just one portion of the protein. We care to describe those states, those transitions, and those functions. We do not care about the portion of the proteins that could be folded or globular proteins, okay? So we do not care to describe functions about folded domains, okay? So we are only focusing on the disorder region of the protein.
Vicky, we have a question.
Yes.
What do you mean when you say transition?
We're going to be looking at it more deeply throughout the course. But what I'm trying to say is, as these are states, these are or might not be stable states in the sense like they are during the whole time, the whole life of the protein maintains a disorder state. There are cases where disorder regions could fold because they are interactive with a partner, because there are conditions like the pH, the temperature just promote the folding of that region. Or the other thing could happen as well. We can have some protein that is folded or seems to be folded under physiological conditions, but then when something changes or DNA jumps into the mix, it gets disordered and gets flexible or gets more characteristic of a disorder region. I hope it's clear enough. Again, we're going to be talking about this a lot during the course. Thank you for asking.
We have another question regarding, is it possible to filter proteins by global or local disorder as a first search?
Well, you could not browse, but you could, when you get into Discord and go to the browser, one of the columns there is going to be disorder content. So if you can sort and get the highly disordered ones on the top or in the bottom, and if you download the information, you are also going to get the disorder content of the entries. So in that way, you can say, okay, if it's 100%, it's globally disorder. And if it's lower, it's just local. It's important to know that in DISPROT, we have the curated data that is experimentally validated. So this doesn't mean that if a protein is completely disordered, but we don't have information, there are not publications, we haven't found them, They are not going to be in this plot yet. We're trying to get there, okay? We need your help to do it. So going back to what an annotation looks like. Again, this is a curated piece of information. Someone added this information. It doesn't matter what kind of annotations we do, whether it's a structural state, transitions, or the functions. As you can see, these are the different kinds. All of the important parts of the core parts, the information that we add, is pretty much the same. So the core information that we add into this product, that you can find in this product, again, relates to publication. So the first thing that we can notice is that we are going to have the reference of the scientific publication where this information came from, with the name and the PubMed ID and cross-link. We're going to also find the information about the method that was used by the authors to assess the information. And what was the assessment about? In this case, what aspect are we describing for this protein? In this case, it's disorder. It's a structural state annotation about how a region is disordered. We add the information about the position, the boundaries of this region. So if this finding was for the region that was 361 to 433 amino acids. So we are now describing and understanding that this fragment of the protein is found by X-ray crystallography as disorder. We also collect statements. These are snippets or excerpts from the publication that supports this finding. Okay? So this is the core information. All of the different types of annotations are going to have this information, but we also have some other optional information that we are always looking to add as well, but might not be available, like different cross-references, as you can find here, or some experimental details, as you can find here. I don't need you to understand right now each and every kind of the information that we create, but just wanted to introduce what an annotation looks like. Okay. Are there any questions so far about this? Okay, I don't see any raised. Okay, so what I wanted to highlight here is that we have a very standardized operation behind this procuration. This is a feature of databases. This is something that databases aim for. We want to provide a standardized way so that we are all talking about the same thing and we prevent or avoid ambiguities, okay? So to do that, we rely on ontologies. So what are ontologies? These are clear definitions that we use to organize and connect the different pieces of information in a database. The idea is to keep consistent the database and the information and easy to interpret and to reuse. As you can see, the terms, for example, that we use are relying on a standard. We try to say every time you are going to find ubiquitin protein ligase binding, we're talking about the same function, okay? So why do they matter? As I was saying before, because we can standardize the terminology that we use. We can reduce the ambiguity and also allows the data integration between different resources. In this plot, we use different things. We annotate different kinds of things. We use different kinds of terminologies or ontology, okay? So for the structural status that we use to say the possible structural state that is populated by a given protein region, for example, disorder. So what is the structural state of this region? Disorder, order, what is this? The structural transitions, to say the possible transitions that may occur between different structural states. This is disorder on this stage, but gets order under a different stage. And then we have disorder functions that are, we call them like this, because these are functions that are very, very, very related to the disorder nature of the proteins. For example, flexible linkers. If you notice, these three first points that I'm making are, say, IDPO. IDPO is our own ontology. It's used in DISPROT to relate to the intrinsic disorder proteins ontology, okay? But then we also relate to other kinds of terms because we also annotate and collect more active, let's say, functions. And for this, we use gene ontology terms. Gene ontology are a huge knowledge base that create terms and organize the knowledge in 3 different groups of functions. We have biological processes, cellular components, molecular functions. For the definition of the experimental methods, we also relate to a standard which is external, and it's the evidence and conclusion ontology. And then we have the experimental details that rely on different control vocabularies. This is the minimum information about the disorder experiment that we add when we feel that we need more information to add to our annotations to provide the best experience for our users and to allow the best understanding of the evidences. So, again, we use IDPO to define structural states, transitions, and disorder functions. You can check what these descriptions and the descriptions of each and every one of the terms if you go to DISPROT.org ontology or to the tab ontology on DISPROT. This is an ontology that we create ourselves, and this is important to note, it's going through some changes because we are always updating and adding more information. So it's possibly going to be changing, hopefully in the next 3 months, but we're going to keep you posted, and the structure is going to remain the same. We will have a structural state, transitions, and disorder function. Gene ontology, as I was telling you before, is a huge knowledge base. It's an external knowledge base. And they describe molecular functions, which are molecular level activities that are performed by a given, in our case, protein region. Biological processes, which are like larger processes that a region or a protein could be involved in and could probably also involve other actors. And cellular components, to be able to define the locations that are relative to the cellular structures in which these proteins perform their functions, or whether these proteins are a structural component of a cellular component. You can check geneontology.org to know more, and you can check the AmiGo or QuickGo to understand more about the different terms. But the thing here is that the knowledge here is organized as a tree. So as we move on the different branches, we're moving into more specific terms. So under molecular function inhibitor, for example, we can find that ATPase inhibitor activity or channel inhibitor activity as child terms, as more specific terms of the one that is in black. The evidence and conclusion ontology is pretty much the same in the terms that it's a tree of terms and we can find more specific terms. In this case, ECO focus on or represent experimental techniques, and we use it to say what methods were used in the different publications to assess the intrinsic disorder. As I was telling you before, we are always welcome to search through the evidence and ontology to find these trees. They are also kind of available in DISPROT ontology. You can go and look for them as well. And we in DISPROT and in any kind of database, really, we try to be as specific as possible. So if you were to find an experiment that is based on a small angle or SAX, small angle X-ray scattering evidence, or SAX, annotating or selecting like scattering evidence wouldn't be wrong, but you could be more specific. So you can go and check for the different trees and check for the different terms that are available in ECO to do your annotation. So, any questions so far? I think not. Okay. It's been a half an hour. I've been pretty fast so far, but I want you all very well rested for the next part. So let's take a 10-minute break. I wanted to say 7, so let's make sure that we are all here in 10 minutes. Go ahead, take care of your biological needs, and we will meet in 10 minutes back here so we have our energies back on, okay? Back to my setting. Okay. I'm sharing still, right? You can see my, let me just, I don't see you.
Yes, we can see everything. Your slide.
Sorry?
Slide.
My slides.
Yeah.
Okay, awesome. Let me put this here. Side, touch this. Okay. Well, let's go into the curation process itself. We now know what this plot is. We know what a displot entry is, what annotations look like at least, and the fact that we try to add as standardized information as we can, relying on different kinds of ontology. So, how do we do curation? Okay, the essential information for curation is firstly a peer review. This is very important. Peer reviewed article with scientific experiments. The proper identification of an UniProt ID, this is how we identify proteins. We need to be sure that we are finding the UniProt accession number or ID that is exactly the same as the one that is being described by the authors in this scientific publication. We need the experimental methods detailed. We need to be able to understand and to properly identify the protein region boundaries, the start and the end position, if the authors are not describing the whole protein, but just a portion of the protein. The region that is being, the most important thing that you have in mind is that the region that we are going to be describing is annotated as disorder, whether this is the annotation and this is the information that is getting the information about disorder for this region, or there's another previously annotated information or description that this region is disorder. Then you can add information about functions, the transitions, or whatever this region or protein do. And then we will need the publication or a curator statement. More on that to come. So the rigorous data creation and the fact that we take so much time with each and every one of these steps and we look into them with very close attention is that we need to have rigorous process to ensure the accurate and comprehensive representation of this protein and of this information in this protein. Because we also want to facilitate the data accessibility and the reusability of the data that we upload and add into this protein. And because we promote the standardized and reliable information that other researchers, other groups, and other databases are going to be using and basing their experiments, their assessments, their theses, or whatever on. Okay, so how is the creation process? What's the layout? You can start curating by two different starting points, let's say. You might find a protein that you find is interesting, and of course, you think it's disorder. And then you look for publications, scientific publications about that protein and about those structural states. Or you find a publication that is describing a protein that is disorder, and then you identify this protein. Either way, the next steps are the same. After finding proper publications with information that are available to curate, you need to determine the correct UniProt accession by checking the construct sequence and the source organism that is described in the publication. Then identify the experiment used to assess the disorder region, identify the different region boundaries, the start and the end position regarding the amino acid sequence, Identify the relevant disorder aspect that you are going to be annotating. This means, is this information the authors are giving me related to a structural state or a transition, or is it a function? And then identify further and specify which aspect of annotation. If it is a structural state, if it's disorder, if it is a function, which kind of function? And then collect all the statements that are present in this publication, all the things that the authors are saying that are supporting this evidence. And lastly, but not less importantly, add all the cross-references and all the additional information that you can, that are relevant at least, for understanding the experiment and the findings. This could be also looking at the experimental setup or the experimental details. So let's go to the first step. Let's try to find IDP-related evidences. What we do, or the strategies that are in hand, include querying PubMed or Europe PMC for publications, Google Scholar, of course, any kind of search engine, for publications with disorder keywords or disorder adjacent keywords. So effective terms could include things like intrinsic disorder, disordered, and structure. And you can also look for things like mobile, flexible, more ADP adjacent, let's say, but still could highlight or show that a region could be disordered. And you can also look for the usual methods or experimental techniques that are related to or are usually used to describe features of disorder regions, like NMR, for example, Frecht, Sachs. There are cases in which authors may describe disorder without using explicit terms. Terms like not visible, for example, or missing electron density are things are very, very, very commonly found when are publications about experiments related with X-ray crystallography or cryo-electron microscopy, experiments. So again, this is kind of examples of disorder adjacent, or not even adjacent, but just hints that could point us towards information about disorder regions. You are, as curators, you are going to get better at finding strategies that work for you to finding these kinds of information, but these are great starting points. So let's say we find a publication. Next, we need to identify the UniProt, the protein, using a UniProt identifier. So the ideal scenario is that the peer review publication explicitly says, okay, we are testing this protein, which has the UniProt accession number Q215, whatever. That's not necessarily the case for all of the publications. There are cases where they're not providing UniProt identifier, but providing other kinds of identifier, like the NCBI gene, the ensemble ID, or other kind of cross-references. That would be like the second best case scenario. And then if we get those kinds of IDs, we can use the UniProt ID mapping service that is linked here in the this is UniProt, for those that are not familiar with it, to map them. So if you get this ensemble ID, which is the UniProt accession number for this, you will get the ID from UniProt regarding that other cross-reference. There are other cases when authors are not going to explicitly provide an ID. They might tell you the nucleoprotein from SARS-CoV-2, and maybe SARS-CoV-2, you need to find which is the protein. You should go to UniProt, look for the sequence that is there. In some cases, authors also provide a bit of a sequence, an image, or whatever. Or even if you are provided with a cross-reference identifier that is different from UniProt, you are always encouraged to do a blast search to make sure you can just blast the sequence and see what blast finds in UniProt. Or if you have like a candidate that you think, oh, I think this protein seems to be like the one that they are describing. If they provide some kind of sequence or information, you can blast them, the two together, to make sure, compare them, to make sure that they are the same. That is the worst case scenario, when the authors don't really say which protein there is, or there are confusion because there are many strains from the bacteria or isolate from viruses or whatever. But they might mention there's a histidine in the 35 position. So you might become a bit of a private investigator in some cases, trying to figure out and find where you can find the information. These are cases that are not very often, don't really happen, but sometimes they happen. If you ever find an issue trying to find the UniProt accession number, of course, you can reach out to our larger community of curators or to us, and we will try to help you. Okay, but most of the time, more so with the latest publications that are there, they're going to have some kind of ID. So it should be easy to find the UniProt accession number. So we have the publication, we have the UniProt accession number identified. Let's go to annotating structural state, transitions, and functions. When you get to be a Disprot curator, you will get a curator account. So when you log into Disprot with your account, you will be able to get into different entries, and below the feature viewer and all the things that we were saying before, you will find the curation interface that looks something like this. If you pay attention and remember all the different core information that we have on the annotations, you will see that it's basically the same thing. This acts as a form for you to fill out with the information that we want to collect. Reference identifier, the Ecoterm or the methods that was used by the authors, boundaries of the region that is being described, the start and end position, the aspect, what are we annotating, cross-references, statements, and then we have below some additional fields that are related to the experimental details. Okay, so let's go to the top. The first thing you are going to be asked for is the reference identifier. In here, you need to add the PubMed ID of the publication that you want to annotate. Then we'll have the Ecoterm and the methods. You don't need to know all the Ecoterms. You don't need to know the codes, okay? You just need to know what kind of experiment it is. So if the authors are describing a circular dichroism experiment, you will just come here and type cellular dichroism, and Disprot is going to retrieve all the terms that are from ECO, from the methods, that relates to circular dichroism. So now we can find, again, as a tree representation, where we go from the broader term to the more specific terms, the circular dichroism evidence using manual assertion, but then we can find far UV circular dichroism, near UV circular dichroism, and even synchrotron radiation circular dichroism. You're encouraged to find the most specific term. So if the authors are describing a far UV circular dichroism, it won't be wrong that you annotate this information or select this circular dichroism evidence, but it will be better, and you will be encouraged, and you will probably hear from us being like, oh, could you please change it to a more specific term like far UV CD? Once we add the methods, we are welcome to the next step, which is adding the boundaries, the start and the end position. The very important thing is that we need to add the start and the end position regarding the amino acid sequence as per corresponding to the UniProt entry. So this means sometimes let's say that we have a protein that has 200 residues, okay? It's 200 amino acids long. The authors are just assessing from the 100 to the 200. So they just create a peptide, just assessing a fragment. And in the publication, they might say, okay, we assess by circular dichroism or by NMR or whatever, this peptide, and we found that the first 10 residues are disordered. Here, when you're asked about the boundaries, you should not say the start is 0, let's say, or 1 residue, and the end is 10. We are not talking about the first 10. We're talking about from the 110, from the 100, residue to the 110. So this is very important that we are always related, not to what was the experiment, but what this is telling us about the protein. Okay, I don't know if I'm being clear. Is there any questions? I could probably rephrase this way better. Okay, I'm guessing not. But the idea is, okay, we are always relating to the UniProt accession, the UniProt sequence, okay? So once we have the reference identifier, the method, and the boundaries, we're going to move forward to the different aspects. What are we annotating? What kind of evidence are we annotating? Okay, we have 4 different options. We have the structural state, structural transitions, what we call disorder functions and use IDPO, and the more active function or gene ontology function. Once you select, let's say we select structural state, so let's go and see how we do structural state annotations. For our annotations, we have different length requirements. Why? Because it doesn't mean anything. If we were to say, okay, these 3 residues, 4 residues of a protein are disorder, it doesn't provide much of a biological information because 4 residues are not able to form any kind of a structure. They're always going to be like flexible and just elongated. Okay, so we want to collect in this plot what's biological relevance information. So we have a length requirement for the structural state of about 10 residues. So anything that is beyond 10 residues, you can annotate as disorder, for example. Okay, so within...
Sorry, Vicky.
Yeah.
We have a question.
Yeah.
How do you set the boundaries if you have more than one IDR in your protein?
You will be creating different annotations. You will create one annotation for the first IDR and a second annotation for the second one. Okay? This is very good question. So thank you, whoever that asked it. Every annotation relates to a region. Okay? It could be about the structure or function or whatever, but we are always discussing or providing information about a certain region. Okay, so any other questions? No? Okay. So again, when we are annotating a structure state, beyond trying to say, okay, this is information about the structure state of a region or a protein, we need to further specify and say, and define the term or be the more specific as we can regarding the aspects. So most of the annotations that are related to the structural state are going to be about disorder. But if you look into what we have in this plot, if you select a structural state, you will also get a tree in which disorder is like the parent term or like a larger branch in which we have other intermediate or more specific terms. This is pre-molten globule and molten globule. Why? Because disorder or proteins are not just disorder or just order. It's like a spectrum, of course. And there are different stages and different intermediate states. You can go from the farther from the spectrum on the more disorder side, from the extended conformation, going through the gain of secondary elements and even tertiary elements to a folded, fully ordered and structured domain or protein. So we not only collect the most extended regions, but we also collect those that are in between. Disorder works as the term disorder works as the general term, okay? But if we find that the authors are saying that a region is pre-molten globule or molten globule, you are more than welcome to further define the region as one of these. Pre-molten global is defined as a more condensed state. It's not fully extended, it's more condensed, and it has some residual secondary structure. It could be transient, it could be a weird shaped helix or something like that. And then we have, when it's a bit more condensed, we have molten global. It's a more compact state than pre-molten and may have native secondary structure, but lacks tertiary structure. Under our definition, well, not ours, but like IDP community definition of what disorder is, we also consider disorder when there is secondary structure, but there's no tertiary structure. So this is not a globular protein or globular domain. This is also a kind of disorder, okay? So whenever you find these intermediate states, you are welcome to add them and to be very specific. And then we can also find that there's another term or another possibility to add information about order, the other very extreme case on our spectrum of structural states. Order, of course, as you know, is the compact state with stable 3D structure. And you might ask me, why do we annotate this? If I said before, I'm going to say it again. We are a database of intrinsically disordered proteins. We care for adding information about the regions of proteins that are disordered, the structure state, the functions, whatever, but the disordered regions. We do not care about adding information or describing regions that are folded or globular proteins. So why? Okay, so you have it right here in yellow. Order or structure regions or annotations should only be annotated or added when they represent a state as in sometimes it is ordered, sometimes it might not. And that evidences or highlight the fact that the region is part or goes through different structural transitions, okay? As we were talking about before, there are sometimes nothing in biology, or very low, very small things, or little fewer things are stable in biology. We are always, it's a very dynamic thing. Proteins that present disorder region might, under certain circumstances, get folded. When this happens, we are interested in those kinds of information. So we have the order annotation to highlight those cases in which the protein gets the structure, okay? Another very, very, very important point here that I wanted to make is intrinsic disorder is not the same as denaturation. Because I was here talking about how, depending on the context, maybe an order region could go to disorder or a disorder region could go into order. But this doesn't mean, like, if you find a publication that the authors are saying, oh, we are looking into this globular protein, I don't know, whatever it is, phosphoprotein that is structured, but when we put it at 95 degrees Celsius, it gets very extended and has characteristic of unfolding. This is not intrinsic disorder. This is a case of denaturation. So please do not consider experiments under extreme non-biological relevant conditions, because every protein is going to be unfolded. No, it's not unfolded. Well, yeah, unfolded if you put them under extreme conditions like pH, temperature, or whatever, okay? We care for the features that are intrinsic, related to the proteins, and biologically relevant. Any questions so far regarding the structural state annotations? Okay. Now, let's move on. We were talking about this a bit before. Let's move on to structural transitions annotations. In this case as well, we have the length requirement of about 10 residues. Anything below 10 residues, you are not going to be able to annotate as transitions. So have that in mind. And as I was saying, transitions could happen between disorder to order states and order to disorder state. I'm sorry, did I say the same thing? From disorder to order state and from order to disorder state. Going through all of the kinds of disorders Okay, so all of the different intermediate states could go or be subject to structural transitions. So if you go into this plot to the annotation interface and you select structural transitions, you will see all of these different kinds of terms available for curation. You are going to find the very broad term and very okay to use on curation, disorder to order, but you can also find disorder to multi-global disorder. So it's a disorder state going into still a disorder state, but a bit more compact, or pre-multi-global to multi-global, pre-multi-global to order, and the same for the other arrow, the other sense, from order to disorder, and the more specific terms, if there are transitioning between intermediate states, like from order to multi-global, for example. You are also welcome to specify the source state and the target state. You see here, I'm sorry, here. If you were to say it's a transition from disorder to order, you can add here cross-references to other annotations about the disorder state or the order state, quite like this. So if this is annotation about a structural transition, again, these are like purple-ish annotations. We have disorder to order, and then you see here, okay, besides all the points and all the information that we already know that annotations have, we have that this is a transition between this source state and this other source state. If we look into it, the first one relates to a disorder annotation that was previously added by our curator, and the order, the target there, is related to an order annotation. This is why we have this order term, to be able to add information about when, why this is happening, why this region gets ordered. Okay? Any questions about transition states or what transitions are? Yep. Okay, I'm going to move forward then. So now we moved into the function annotations. Firstly, we're going to go through the disorder functions. We call them because these are terms terms that are in IDPO, in our own ontology, and this is because they are very, very, very related to the intrinsic disorder nature of the protein. So in this case, for functions, we have a lower length requirement. You can add information starting from 5 residues. This is because there are main motifs, or like you want to say, okay, these 3, 4, well, actually 5, 6, 7 residues are the ones that are binding or doing any kind of activity or involved in any kind of activity. So within IDPO, the disorder functions, as you can see here, we have 3 major branches or cases. We have entropic change functions that also have like more specific terms like flexible C terminal tail, flexible N terminal tail, and flexible linkers. And then we have molecular recognition display sites that have very long different terms that relate to PTMs, like whether a region of the protein is being is serving as a display site for post-translational modification or PTMs, like glycosylation, phosphorylation, acetylation, whatever. Yeah, have a long list there. And then we have some self-regulatory activity terms. Since disordered proteins are very flexible and very promiscuous, there's sometimes that they can interact with their own, with other regions within the same protein. So this is not dimerization, this is not polymerization or any kind, this is just self-interactions. They could be like self-assembly or self-binding when they're like just one portion of the protein is binding to another, or this interaction could have a different kind of outcome when they are regulating the activity. For example, when one portion of the protein is working as self-inhibition or self-activation, maybe a region of the protein is preventing another part of the protein folding correctly, or a pocket being available for joining with a partner or whatever, we can add that information as, for example, self-inhibition. You can find in this .org all the different definitions, the different terms, for example, and the different definitions that each and every one of the terms have. So if you have any questions, if it's not clear, whatever, you can go and check, and of course, ask us, for sure. Important to note, yeah.
Sorry, Vicky, we have a question. It's related to the transition part. Is it necessary to make an entry between every two different states? For example, between pre-molten and molten globule, or between molten and random coil, and so on?
No, it's not necessary. It's if you find a region in the same publication or even in different publications. Let's say you find a region that under certain conditions is disorder, and then on another condition or wherever they found as pre-molten. I don't know if you meant to link them or to put them both. I'm going to try to ask or address both potential questions. So if you find the two pieces of information, we try to add them both, because there might be some context or important information that is important to have to say or to understand how the region behaves under certain conditions or whatever. So if you find the different evidences, we will ask you probably to add them, or if we see that it's missing, we'll probably add it ourselves. Regarding the transitions, it's not mandatory. Normally, when we add transitions, is when the authors are clearly stating there is a transition in a way. So if you were to find like these two states but no link between them, it's not necessary. You could add it, but if it will be different if in the publication they're like, okay, this is completely extended, completely disordered, but when it's binding to a different partner, it becomes more compact and gets pre-molten. That would be a great idea to add that as a transition. It's never mandatory, but it will be best. Because when we are adding information into this, but we try to aim for completeness. So we try to, ideally, create all the information available on the protein. It's a hard task. But then when we are looking into publication, all the information that we can find from the publication. So again, not mandatory, but very appreciated, let's say. Okay. Any other questions? Okay, awesome. So we check structure states, annotation, transitions now, and disorder functions. Again, disorder functions are those that are really, really, really related to the disorder nature. As I was telling before, all of these cases rely on IDPO, intrinsically disordered protein ontologies, and we are doing some changes on it. So you might not see this exact layout when you go into this. I'm just mentioning this, you're probably going to forget, but I'm going to mention it, so just you're aware, okay? You might see a bit of a difference on the layout here. It's going to be could look something like this, where you'll have different kinds of terms, but as you can see, you won't have the add button. So the available terms for curation, what they mean, what they stand for are the same, okay? You will have the entropic chain, different terms, the molecular recognition display site, and the self-interactions. Whenever we add more terms and we have more classifications or wherever, we're going to update our ontology page. We are going to let you know. We're going to, do not worry. But just to be, I just wanted to flag that you might see something slightly different, okay? But the same as I was talking about before. Okay, so let's move into the gene ontology functions. Again, these are more active, let's say, functions, okay? The length requirement here as well is a bit lower, it's 5 residues. So functions you can annotate for regions that are from 5 residues to longer regions, of course. We use the 3 different branches from gene ontology: molecular function, biological processes, and cellular components. And of course, you can use Quick Go and Amigo, which are two tools. I personally use Amigo, which is here linked for your consultation, for efficient annotation and in-depth understanding of the terms. This means you can go into these pages to see the tree layout to understand whether there are more specific terms or what you might need. You can type in, this is a bit of a still screen, screenshots. You go into Amigo and you can type like something here like protein binding, and they're going to retrieve all of the different terms that they have regarding protein binding, and of course the synonyms and the definitions. So it's very important that before selecting a term or whatever, you double check and make sure that it's the most specific one you can find and it's applied to what you are trying to annotate, okay? Of course, again, as the same we were talking about with methods and eCode, you don't need to know the exact names or the codes, okay? If you go and select function geo from the aspect, and you can start typing, for example, viral, and the interface is going to retrieve all of the functional or the most relevant, let's say, gene ontology terms that has viral as part of the name. You can keep on typing and the search engine is going to get better. So if you just type viral factory, it's going to retrieve viral factory, okay? Another important point is that when we are adding gene ontology functions, depending on what kind of functions, we are also sometimes asked to add additional information like interaction partner, for example. So if I were to select protein binding, then I'm going to be asked, or I'm going to see this field that says interaction partner. It could be protein, it could be DNA, it could be many, many things, okay? So you can select different databases to identify this partner. For the case of proteins, we're going to select UniProt, and we're going to add here the identifier, the UniProt ID. We can even further specify which region of the protein is the one that is the partner, in this case, of the binding. But we could also be like DNA binding and select an NCBI gene, for example, or RNA and select RNA central and an identifier from it, okay? This is considered one of the cross-references that we are encouraged to add every time we get, okay? Any questions about gene ontology functional annotations in this script? I know this seems like a lot of information, but trust me, the interface is going to be helping you get the best terms that you can, okay? Okay, let's move on. We are so close to be done with the different annotations. So then, this is, as I was telling you before, this is one kind of the cross-reference, is the identification of the different partners, but we have other kinds of cross-references. We can add information or cross-reference other external experimental data databases. Depending on the method or assessment that the authors and the publication is referring to, you can find different resources that could potentially have deposited data. The authors are the ones that should deposit the data on these other resources, depending on the type of experiment. So X-ray crystallography, NMR, cryo-EM data are often deposited in PDB, Protein Data Bank. BMRB serves as a deposition for NMR-related data. Then we have SAS-BDB for SACS and SANS experiments, EMDB for electron microscopy data, and PCDDB for circular dichroism and SRCD data. Sometimes the authors in the publication say that the data is deposited in blah, blah, under this cross-reference. That's great. We are very happy to see those things, those cases, because all of the information that we can add to our annotations is the best, okay? Sometimes authors deposit the data, but they don't explicitly say that in the manuscript, and sometimes they don't deposit the data, okay? But if they do, we try to find them and to cross-reference it there. And then, lastly, we have the statements. The statements are small snippets, concrete. We don't want to paste the whole publication, that are part of the publication that supports this information that we are annotating. The idea is that users or other people can go into this product, look at the annotation, and get the sense of the highlighted information about what was found, how was it found, and what the authors are saying about it. So to also they can decide how strongly they want to rely on that evidence or not, or there are other things, whatever. We want to provide information. We don't want to crowd the information, but we want to provide all the necessary information for the proper understanding of the evidences. When we try to add statements, we are also asked to select the source. Which section of the publication was this information in? Was it in the abstract? Was it in the results? Was it in the discussions? Most of the useful statements, let's say, are from the results, because there is that oftenly they relate to the experiments. They say, because of the NMR findings, we said we can find that this is disorder. That would be a great statement. But there are cases where the authors just don't say anything. Well, they don't explicitly say this is disorder, for example, or there are statements that we can collect that are not super clear and we want to be more clear on something or whatever. There's also possible to add a curator statement. This is like, of course, we prefer results and prefer, any kind of statements from the publication, not necessarily results. But if you need to add more information or whatever, you are welcome to do it by using a curator statement. This is an original creation that our curator wants to type in or to provide more information about, okay? So let's go to the last portion of the things that we want to add when we are annotating information. And this is the other. It's the minimum information about the disorder experiment. Are there any questions before I get into this regarding the structure state, the transition, the functions, whatever? Cross-references? No? Oh, you can ask after as well, of course. But then we are going to move forward. I'm going to drink a mate, and we're going to move forward to Miade. So, when I was showing you the curation interface, I told you that below we have the additional fields that are related to experimental details. As you can see, we have 3 different kinds of information here that you could add when you are curating information. Let's see a bit about what is Miade before we get into this. As I said before, this is minimal information about the disorder experiment. These are guidelines that were defined by the intrinsically disordered protein community to facilitate the definition and the interpretation, actually, of the results of an IDP experiment. Okay, the idea is that we avoid ambiguity and we provide the most information to do a proper understanding of the evidence. Okay, because sometimes, because actually the nature of the disorder regions and the disorder proteins, they are so flexible, they are so exposed to the environment that they're really liable to the different contexts. This context could have many shapes and forms, could be conditions like pH temperature, but could be other things as well. And they could have really high, hard consequences on the state or the functionality of these regions. So it's really important that we understand what is happening, what's the context of an experiment, to properly understand what is going on and to give the biological context of what the findings are. So sometimes, most times, we don't assess the protein in the physiological condition, ideal conditions, in where like it's the full protein with the exact sequence, amino acid sequence at pH 7, at 37 degrees, just the protein alone. And there are cases when we do things to the sequence construct, we modify it, we add tags, we add any kind of additions or remove any kind of things that could alter the result. Okay, so the kind of information that we want to have and to add in or to be looking at when we are looking into these other experiments is, did something happen to the experimental construct? Is it not the same as the wild type sequence? Is there anything weird there? Or were the experimental sample different? It's not just the protein, maybe there are other components as well. Maybe there's DNA there, maybe there are other proteins. Or maybe there's some conditions that are different from the physiological or different from the standard conditions. Maybe it's not just the method, but just like the pH, it's the temperature. So we want to understand all of these different points to properly identify and to infer accordingly the structural state or the functionality of these regions. Okay? So we do this again by adding information about the three different components of MIADE: sequence construct, experimental conditions, and experimental components. We're going to see what kind of information we can add when we are trying to add information about the sequence construct. In here, we are able to add information about the difference in the protein sequence that the authors may have, or the experimentalists had to do to the protein. So the assessed protein is not exactly the wild type or the one that we can find in UniProt. Maybe they have PTMs, maybe there are non-standard amino acids there, they could be tags, labels, or dyes that are related to the experimental approach, and even mutations. Once you select the different kind of construct, here you have construct alteration. Here also, I don't think I mentioned this, but you can also add the exact sequence, like the amino acid sequence that was actually assessed by the authors. And then you can, this is not mandatory, but it's optional. And then you can add, if there are construct alteration, here, for example, you can add mutation. For each and every one of the different cases, you are going to be asked to further specify. So let's say you selected PTM, you're going to be asked which kind of PTM. You will have a long list of options like phosphorylation, acetylation, glycosylation, for example. When the sample, the protein that is being assessed, has this kind of modifications. Okay. If you were to select tag, for example, you are then going to be able to further specify and say which kind of tag. Is it strep? Is it his? And the location of it. Is it on the C terminal of the peptide of the protein that was assessed, or the N terminal? Okay. And for the mutation as well, for example, you are going to be asked what kind of mutation. Is it an insertion, a deletion, a substitution, for example? And then further identify or specify which kind of substitution it is. In this case, we use HESV nomenclature for this. This has this shape. So it's like a P dot the residue that is being replaced, the position, and the residue that is being replaced by. So in this case, the serine residue at the 272 position was replaced by a cysteine, for example. You don't need to understand every single point right now, but this is just a way to show you all the information that you should be focusing on or be paying attention when you're reading a publication, because you should add them like this, okay? Any questions on sequence construct information? That's all. Then, second point, we can add experimental conditions. These are information that you can add regarding the pH, the temperature, the pressure, and even the oxidation reduction potential. After selecting the experimental condition, you can also select the values and the units. So let's say I said, okay, I'm going to add information about a temperature that was used. I'm going to be asked about which value, of course, the value, like let's say 55. And the unit, is it Celsius? Is it Kelvin? What is it? And the same for each and every one of these other points here. Something that is really, really, really important for me to highlight now at this point is that these fields that I'm showing you should only be used when experimental conditions are essential for the correct interpretation of these experiments. Adding details like this experiment from an E. coli protein was done at pH 7, sorry, at 37.25 degrees Celsius is not recommended. Why? Because they ended up like crowding the information. We want to provide the information that to be for users to be like, watch out for this. Okay, so it makes sense to add if we are looking into, I don't know, let's say it needs to have biological meaning. If we are looking or annotating a protein that is a heat shock protein, for example, or whatever, we are going to find information about that said protein at 37 degrees, for example, and maybe another information of what happens when the same protein is at 60 degrees. In that cases, the information about the temperature is very important because it's probably going to behave, the protein is probably going to behave differently. This is the kind of information that we want you to be able to annotate and to bring. Or maybe a protein, it's moving around between different components in the cell and have different pH or whatever. This is the kind of information that we want. Okay, this protein behaves like this one is in pH 7 and behaves like this one is pH 5, for example. So this is the kind of information that we need. We need to be providing information that other people are going to be reading and say, now I understand what's going on because there's a different pH, because there's a temperature, or you are assessing a protein that is from Extremophilus. Yeah, of course, it makes sense for you to say, okay, this is being tested at 70 degrees or 80 degrees or whatever. Okay? Just wanted to make that point very clearly. Are there any questions regarding experimental conditions, annotations, or information? No. Okay. We are really, really close. Bear with me. A couple of more minutes. The last piece of information that we can add related to the experimental details are the experimental components. This is how the sample that it's being assessed by the author. Are they other interactors or actors part of it? So we can specify if there are interacting antibodies, lipids, membranes, nucleic acid, proteins, small molecules like ions, or even if the experiment was conducted within a cell line. So we know that there might be other actors, or we are not sure, but we cannot actually identify or say which actors they are. So for this case as well, if you were to select, let's say, interacting protein, then you are going to be asked to further identify this partner. Say, okay, cross-reference to a database. If we're going to be cross-referencing to say that there's another protein present in the sample, we're going to use, for example, UniProt. And then we can add the identifier here. We can also add information about the concentration, the unit, and the deviation. We can add information, again, about nucleic acids or whatever. So for each and every one of the different interacting actors, we have different databases that we can use to further identify these partners. Okay? This is very, very important. Mostly, you are going to see these kinds of information mostly on transitions, for example, because there's this thing for intrinsically disordered region that is called folding upon binding. There are many, since they're very promiscuous again, and they have many interactors or partners, disordered regions often folds upon binding and get order transition state because they are interactive with a membrane, a nucleic acid, a lipid, or whatever. So you're going to find these kinds of information a lot in transitions, okay? But this doesn't mean that you cannot find them in other kinds of annotation like structural state or functions, okay? If you find, for example, the authors are analyzing a protein complex, let's say we have, I don't know, calmodulin binding protein that it's been assessed with calmodulin being binding, but a region is disordered, you will annotate this as the region of the protein being disordered, the boundaries and everything, and you will say that this is interacting with calmodulin, okay, for example. So this is how we get, after all this information is added, and all these things are being considered, to a fully annotated evidence. This is the same piece of the same image we were checking of the first slides. Okay, this is, for example, a function annotation. Let's see the parts again now that we know what we are looking into. This is a function annotation again, where the curator was curating a functional function. So this is an active function using gene ontology terms. The term of the activity is ubiquitin protein ligase binding. It was found for this region of the protein. Sorry, I forgot to say which protein this is. I think it's TRIM 2025, I'm not sure. So they found the ubiquitin protein ligase binding activity for this particular protein region, 32 to 37 residues. They found this evidence by X-ray crystallography evidence. They also added the interaction partner identified with this UniProt accession number. And they found this information in this publication. You have the name here and also the link here. And even they found some cross-references to external databases. We have the experimental data like PDV and even a motif. They are cross-referenced here. Okay, they have the different statements from the publication. As you can see, it's possible to add more than just one statement. In this case, the curator added a couple of statements from the results and wanted to be more clear about something and also added a curator statement. And then we see the experimental details or the MIADE details that are related to this function. We see that the protein was modified because they had two residues, 33 and 37, phosphorylated. So the region that is phosphorylated is the one that is binding with this ubiquitin protein ligase. Okay? And we can also see that there's another protein present in the sample as well. Okay? Are there any questions? We're going to take a break now because we all deserve it. But are there any questions about all the different information that we curate, how the annotation looks like, what to do? Okay. Let's take, is this 12:45 already?
Yes.
Okay.
Yeah. I was wondering, about the thing that you said.
Yes, the time, definitely.
What you are missing?
Apologies. So once you get, what happens after curators add a new piece of information? This is the process of curating a new piece of information. Curators get a scientific publication, which they interpret and add the information into the DISPROT entry. After adding the information, one of our reviewers is going to check if the information is okay or not. If it is wrong, it's going to get rejected. This doesn't mean it gets eliminated, it's just going to get rejected and not going to be shown. But if it is okay, it's going to be validated. It's like a double check that everything is okay. Could also happen that the information or the annotation needs minor review or minor edits. So if that happens, two things could happen. The reviewer could ask the biocurator, provide feedback and ask for the modifications so they can go back to the annotation and do the changes and then go back to the reviewer for their assessment. Or the reviewer can edit the annotation themselves. And if there are like small details, so after the edit, it will be like edited and validated by this reviewer. All the pieces of information that get to be validated or double checked are going to be in line for the release that we do, again, every 6 months. Interface. So after the course, what's going to happen after in the next few minutes when we finalize this? After the end of the training, you'll get a short, very simple and easy quiz that you will need to pass for us to open your curator account. It's really easy. You will also be asked to provide some basic information from you, like your PI, who is your PI, the affiliation or where's your university, where do you work in, and your ORCID account. If you don't have an ORCID account, please create one. We're going to remind you when we send the email with the quiz and everything. But because we want to credit you and for you to have your Appicuron account set and ready. And we will also, at some point, not now, but in a couple of weeks probably, going to ask you to provide feedback from this training so we can improve and be better next time, okay? Once you become a curator, you will be added, if you're not already there, in our mailing list. You will be invited to join our Slack space, which is where we communicate general things, we share materials or whatever, and we're going to share materials with you to help you start your curation journey, let's say protein lists or publications for you to curate, okay? Once you do all of these, we do all of this, please go and we open your account, please go to Appicuron and set your profile to public. This will get you the best access to the different features. So you have to do it by going to your dashboard, edit profile, and set the privacy and to public. If you don't do it, you are not going to be able to see some features regarding the badge that we were just talking about, and you won't be able to track as well how's your progress into getting to the different milestones for the acknowledgement in the publication, for example. Of course, if you have any issues with this, you can send an email to us, talk to us in Slack, whatever, okay? Couple of more slides and I will let you go, okay? So biocreation and DISPROT, we focus on many topics and we have many projects. Now we are going to be focusing on stress response proteins. This is going to be part of our release on June. But all contributions are welcome. We are interested in whatever you are interested in. So if you want to contribute to any of the other data sets that we have, or maybe you are focusing on a particular organism, you are welcome to join. You are welcome to tell us, or to, even if you have an idea for a new thematic data set to highlight the features of disorder proteins, let us know, okay? And spread the word, because many of you might know people that are not here today, but wants to contribute these kinds of things. So spread the word. One piece of warning though, just so you are aware, when you become a curator, you will be welcome to add information, as Norby was asking. Welcome to add information and annotations into new proteins, new entries, or already existing ones. If it's a new protein, you'll see the curation interface that I was showing before, and will be free to add as many annotations as you want. If it's a protein that it's already there and you try to add it, you will get a message saying that this protein already exists on this spread, and you will get the link for edit the existing entry, as in coming to add information. But you are not going to see the curation interface. Instead, you will see this thing, which is ask for ownership. This is really 2 seconds. We're going to remove this at some point, but right now it's still in place. So you just say something here like, I want to annotate this protein, and we're going to get the message and give you access. Okay, this is because we have like an ownership system right now, and every protein that you create is yours. But to be honest, we want you to add information to everything. So if you ever encounter this, reach us a line or ask ownership with this button, so we can grant you access to add information. Okay, last 2 slides. I'm sorry, I will let you go as soon as possible. We have many trainings, courses in place. You know, we're going to have an advanced level training next week. Many of you sign up into it. If you didn't sign up, are interested, you had too much fun today and want to join us for the advanced, let us know ASAP, please, so we can arrange you also coming to the advanced training. Then we have some e-learning courses. If you know that people couldn't attend or might be interested or whatever, you can point them into this direction. We have Elixir. In the Elixir platform, we have 2 trainings, one in English and one other in Spanish. It's not as updated as the one I show you today, but it's pretty good and makes the work okay. And also in the Elixir platform, we have some experimental technique courses. So if you want to know more about how to use NMR, FRET, or SACS experiments or understand how they're used for understanding intrinsically disordered proteins, I also welcome you and encourage you to come to this link and to see what we prepared there. Okay? I've linked also some interesting publications. You already have publications in the Google Drive, but again, maybe you can use this. I'm going to share these slides after the course. You're going to find that the slides keep going and going. For example, there's a curation example. I encourage you to look into it. It's very, very silly. It has like 5 slides. And when you go back, forward into the slides, it will ask you things and gives you the answer, okay? This is an example of something that I wanted to show you, but I didn't have the time, of course. Related to some things that you might be needing to have in mind when you are curating. And I'm also going to share probably a small video of how to actually do curation, okay? Okay, we found a publication. What's next point? What should we do? What are we going to do? This is going to be part of the advanced training in more depth. So for those of you that didn't register, again, I encourage you to join us. I know this was really heavy on information. I'm really sorry I didn't get the time to do this mock curation with you today, but I hope to see you next week, and I hope to see you in Slack. And if you have any questions, we can stop the recording probably, so you can ask more freely. And we are, of course, in touch. If you are not joining next week, which I hope you are. And of course, thank you very much for those that only joined this week.